Science grows by disagreement. Every new conceptual leap sounds, at first, like an overreach. Atoms were once undetectable philosophy, imaginary numbers nonsense, and the Big Bang a sarcastic insult. We expect nDNA to trigger the same reflex. In fact, it should. Any claim that the latent cognition of AI can be read as a digital semantic organism deserves hard tests, sharp critiques, and attempted refutations. We welcome all the critical reviews. Here is our stance. nDNA is not a metaphor in search of applause. It is a set of measurable objects and falsifiable predictions. It says that belief updates trace paths with curvature, length, and torsion. It says contamination leaves a reproducible rupture signature in late layers. It says memory, drift, and coactivation can be quantified and compared across families. If those signals fail to replicate across models, prompts, and paraphrases, nDNA should be rejected or revised. In short, I believe nDNA asks to be argued with, not to be believed right away. The very foundation of artificial intelligence rests on a borrowed metaphor, the neural network. Yet biological neurons and artificial neurons share little beyond the name. One is a living, electrochemical cell shaped by evolution, the other a mathematical abstraction designed for optimization. The metaphor has endured not because it is faithful, but because it is useful. It provided a shared grammar for computation and cognition, long before precise equivalence was possible. Let us quickly understand how a biological neuron works. Skip it if it is already known to you.
How do nerves work? Are nerves simply the wires in the body that conduct electricity like the wires in the walls of your home or in your computer? This is an analogy often made, but the reality is that nerves have a much more complex job in the body. They are not just the wires, but the cells that are the sensors, detectors of the external and internal world, the transducers that convert information to electrical impulses, the wires that transmit these impulses, the transistors that gate the information and turn up or down the volume, and finally, the activators that take that information and cause it to have an effect on other organs. Consider this. Your mother gently strokes your forearm and you react with pleasure. Or a spider crawls on your forearm and you startle and slap it off. Or you brush your forearm against a hot rack while removing a cake from the oven and you immediately recoil. Light touch produced pleasure, fear, or pain. How can one kind of cell have so many functions? Nerves are in fact bundles of cells called neurons, and each of these neurons is highly specialized to carry nerve impulses, their form of electricity, in response to only one kind of stimulus and in only one direction. The nerve impulse starts with a receptor, a specialized part of each nerve where the electrical impulse begins. One nerve's receptor might be a thermal receptor, designed only to respond to a rapid increase in temperature. Another receptor type is attached to the hairs of the forearm, detecting movement of those hairs, such as when a spider crawls on your skin. Yet another kind of neuron is low-threshold mechanoreceptor, activated by light touch. Each of these neurons then carry their specific information, pain, warning, pleasure, and that information is projected to specific areas of the brain, and that is the electrical impulse. The inside of a nerve is a fluid that is very rich in the ion potassium. It is 20 times higher than in the fluid outside the nerve, while that outside fluid has 10 times more sodium than the inside of the nerve. This imbalance between sodium outside and potassium inside the cell results in the inside of the nerve having a negative electrical charge relative to the outside of the nerve, about equal to minus 70 or minus 80 millivolts. This is called the nerve's resting potential. But in response to that stimulus, the nerve is designed to detect pores in the cell wall near the receptor of the cell open. These pores are specialized protein channels that are designed to let sodium rush into the nerve. The sodium ions rush down their concentration gradient, and when they do, the inside of the nerve becomes more positively charged, about plus 40 millivolts. While this happens initially in the nerve right around the receptor, if the change in the nerve's electrical charge is great enough, if it reaches what is called threshold, the nearby sodium ion channels open, and then the ones nearby those, and so on and so forth, so that the positivity spreads along the nerve's membrane to the nerve's cell body, and then along the nerve's long, thread-like extension, the axon. Meanwhile, potassium ion channels open, potassium rushes out of the nerve, and the membrane voltage returns to normal, actually overshooting it a bit. And during this overshoot, the nerve is resistant to further depolarization. It is refractory, which prevents the nerve electrical impulse from traveling backwards. Then, ion pumps pump the sodium back out of the nerve and the potassium back into the nerve, restoring the nerve to its normal resting state. The end of the nerve, the end of the axon, communicates with the nerve's target. This target will be other nerves in a specialized area of the spinal cord to be processed and then transmitted up to the brain. Or the nerve's target may be another organ, such as a muscle. When the electrical impulse reaches the end of the nerve, small vesicles or packets containing chemical neurotransmitters are released by the nerve and rapidly interact with the nerve's target. This process is called synaptic transmission, because the connection between the nerve and the next object in the chain is called a synapse. And it is here, in the synapse, that the neuron's electrical information can be modulated, amplified, blocked altogether, or translated to another informational process.
To anchor the metaphor in facts, here's a side-by-side snapshot of biological neurons versus their artificial counterparts across the dimensions that matter, what they are, how they compute, learn, connect, scale, and endure. This framing clarifies where the analogy holds and where engineering diverges from biology. Let's read out row by row. Nature of the unit. Biological, a living cell with dendrites, a soma, and an axon. Artificial, a math unit that does a linear step and a nonlinear step. Computation. Biological, spikes and ion channels with rich, nonlinear mixing in dendrites. Artificial, weighted sums pass through an activation function. Dynamics. Biological, runs in continuous time with oscillations and rest periods. Artificial, updates in discrete steps and is usually stateless per layer. Signal transmission. Biological, electrochemical, noisy, and probabilistic. Artificial, floating point arithmetic that is exact and deterministic. Learning rules. Biological, local plasticity such as Hebbian learning and spike timing rules. Artificial, global backpropagation with gradient descent. Connectivity. Biological, limited by 3D anatomy and wiring costs. Artificial, arbitrary fan-in and fan-out, often via dense or attention links. Scale. Biological, about 100 billion neurons and 100 trillion synapses. Artificial, millions to hundreds of billions of parameters in large models. Energy. Biological, roughly 20 watts for a whole brain. Artificial, training can use megawatt-scale data center power. Evolution and adaptation. Biological, shaped by survival, evolution, and metabolic efficiency. Artificial, driven by objectives and data distributions. Robustness. Biological, highly fault-tolerant with lots of redundancy. Artificial, can be brittle to adversarial noise and can forget catastrophically. Information storage. Biological, stored in spiking patterns and synaptic strengths. Artificial, stored in weights, embeddings, and latent vectors. Purpose. Biological, survive and adapt in the real world. Artificial, optimize for tasks like translation, dialogue, and search. In conclusion, the neuron metaphor is useful, but it is not the whole story. Brains are living, continuous, noisy, and shaped by evolution. Neural networks are numeric, discrete, precise, and shaped by objectives and data. That gap explains both why AI scales so well and why it fails in brittle ways. The scientific community uses the metaphor to guide intuition, then switches to engineering, where it diverges. For neural genomics, this means borrow what biology gets right, local plasticity, redundancy, and robust signaling, but measure models in their own native terms, weights, activations, geometry, and data. If we keep both halves in view, we can build systems that are not only powerful but also traceable, resilient, and trustworthy. This parallel is instructive for nDNA. Just as neurons in artificial networks are not literal neurons, nDNA is not biological DNA. It is a mathematical framework that borrows the grammar of biology, lineage, mutation, inheritance, pathology, to describe something real but difficult to name, the latent geometry of model belief. Critics may argue that such borrowing risks anthropomorphism, but history shows that science often begins with metaphors that sound poetic, only to crystallize into precise technical vocabulary once their utility is proven. Indeed, terms like imaginary numbers, the Big Bang, and black holes all originated as linguistic overreaches before becoming indispensable pillars of mathematics and physics. Read the table left to right. The left column reminds you what a genome does in living systems. The right column shows the nDNA analog inside AI. Basic units become geometric primitives like curvature and length, mutations become spectral distortions or torsion, recombination becomes model merging, gene expression becomes trait activation such as multilingual drift or tool use. Let's read the comparative table row by row. 1. Basic unit. Biological DNA, nucleotides A, T, C, G. Neural DNA, geometric primitives, curvature, length, gradient. 2. Information encoding. Biological DNA, genetic code that specifies proteins. Neural DNA, latent geometry that encodes priors and beliefs. 3. Inheritance. Biological DNA, passed from parent to offspring during reproduction. Neural DNA, transferred across model families via pretraining, fine-tuning, and distillation. 4. Mutation. Biological DNA, base substitutions, insertions, deletions. Neural DNA, spectral distortions, torsional reconfigurations, loss of thermodynamic length. 5. Recombination. Biological DNA, crossover during meiosis. Neural DNA, model merging, neural marriages that create hybrid latent genomes. 6. Gene expression. Biological DNA, differential activation by environment. Neural DNA, emergent trait activation, multilingual drift, tool use, task specialization. 7. Epigenetics. Biological DNA, regulatory marks that change expression without altering sequence. Neural DNA, alignment, instruction tuning, or quantization that change behavior without changing core pretraining. 8. Pathologies. Biological DNA, oncogenic mutations, viral insertions, heritable disorders. Neural DNA, alignment scars, scar, adversarial infections, viral, stealth poisoning, nephos. 9. Repair mechanisms. Biological DNA, DNA repair enzymes and redundancy in coding. Neural DNA, fine-tuning, adversarial immunization, safety alignment protocols. 10. Evolutionary drift. Biological DNA, small changes that accumulate over generations. Neural DNA, semantic drift across repeated fine-tunes, cultural biases, or model collapse. 11. Fitness landscape. Biological DNA, adaptation for ecological survival. Neural DNA, optimization for tasks, benchmarks, and alignment criteria. 12. Molecular carriers. Biological DNA, double helix of deoxyribonucleotides. Neural DNA, high-dimensional manifolds of latent vectors. 13. Observable phenotype. Biological DNA, organism traits and functions. Neural DNA, model outputs, dialogue style, translation, reasoning patterns. 14. Genomic sequencing. Biological DNA, decode nucleotides to reveal the blueprint. Neural DNA, extract nDNA to reveal the latent epistemic identity of models. In conclusion, this is not biology. Neural DNA is not a molecule and it does not copy itself in a cell. It is a mathematical object measured by 3 families of signals: spectral curvature, thermodynamic length, and belief gradients. Together, they trace the shape and motion of models' latent belief. Where biology talks about mutations, we see torsional instabilities. Where it talks about inheritance, we see biases carried across pretraining and fine-tunes. Where it talks about drift, we see slow semantic divergence. By reading these signals as a genome, we get practical power. We can diagnose contamination, audit alignment scars, compare families, and forecast failure before it shows up in outputs. nDNA turns a black box into a tractable map, not by pretending models are alive, but by giving their inner cognition a precise geometry we can measure, govern, and improve. If we look back in recent past, we see how Hinton, Bengio, and Lacan put a deliberate push for the term deep learning. Why this matters? In the early 2000s, neural nets carried decades of baggage. To revive representation learning and reset perceptions, a small group led by Jan Laken, Jeffrey Hinton, and Yoshua Bengio made a deliberate rhetorical and scientific pivot. They rallied the community around the banner of deep learning and backed it with concrete algorithmic results. The timeline in fast forward. 2003 to 2005, reframe. Push deep representation learning and rally people around a simple name, deep learning. 2006 to 2007, prove it. Layer-wise pretraining and deep belief nets show how to actually train deep stacks. 2009, codify. Bengio's monograph lays out the theory and the recipe for learning deep architectures. 2015, consolidate. The Nature review by the trio defines the field and its open problems. Why the words mattered, the name simplified the mission, reset the conversation, and gave the community a target to build toward, while the papers made it real. What this means for neural genomics, neural DNA borrows this philosophy from history. A crisp name, a unifying lens, the geometry of latent cognition, and a growing stack of reproducible measurements and mathematical measures. The aim is not branding, it's coordination. Just as deep learning organized a scattered set of ideas into a program of research, nDNA organizes contamination diagnosis, alignment forensics, and model lineage into a tractable science. Clear language first, cumulative evidence next, then the field follows. We are following the path what history teaches us. Let's watch a clip from Laken's recent interview, when he recalls how they revived the area with the term deep learning.
The first one, interestingly, was a system that was deployed in ATM machines, where you can deposit a check and it kind of reads the amount on the check. We knew this had a lot of applications. What we did not realize was how complicated it would be to convince other people that this was so good. It was very difficult to reproduce. Everybody was using different computers, different operating systems, different programming languages. We had something that worked, but we could not really kind of share with other people.
What then turned actually deep learning into mainstream technology?
And in 2003, I became a professor at NYU. I restarted a research program on neural nets. I said, those techniques really work, and we need to revive the interest of the community into those methods by demonstrating that they work. People were making fun of us, people who were working on neural nets, and we started changing the name. Because neural net had a bad reputation, and really was kind of a little too restrictive for the kind of stuff we were doing, we changed the name and we called it deep learning.